In this lecture, we're going to take a look at the cardiac cycle and how we control the cardiac cycle using the cardiac conducting system. So let's take a look. So the muscle of the heart can be either contracted or relaxed. Systole, and that's how it's pronounced, you actually say that E at the end of systole. Systole is contraction of either the atria or the ventricles of the heart. And when the atria contract, which is what we're seeing here, is systole of the atria. As the atria contract, that pushes blood from the atria into the ventricles. At this point, the ventricles are in diastole because they're relaxed. And that is the time when the chambers of the heart are able to fill with blood. So relaxation is diastole, contraction is systole. And if you think about the names for blood pressure, so let's say if ideal blood pressure is 120/70, this 120 is called the systolic pressure, and the 70 is the diastolic pressure. And what we're measuring is the pressure that is being generated by the ventricles when we measure this blood pressure of 120/70. So let's take a look for a second at what's going on in the second picture. Here, so if in this one, the atria were in systole and the ventricles are in diastole. In this image, we're seeing now the atria in diastole. They're relaxed and they are filling with blood. And the ventricles down here are in systole. So you can see here they're relaxed. There's a lot of open space inside the left and the right ventricle. But here they're contracted, so the open space is much smaller. So we're generating pressure that pushes blood up and out through the pulmonary trunk or up and out through the aorta. So as we make this pressure here, especially at the left ventricle, then the arteries beyond are being pressurized as well. So when the left ventricle is fully in systole, it generates a pressure of about 120 on a healthy person. When the left ventricle is fully relaxed, the pressure is significantly reduced to hopefully 70. I think more people on average, that number is 80, but what we want to see is a little bit lower than even 80 in when the ventricles are in diastole. And that would indicate our Red just joined us, as I'm sure you probably heard. What the goal is to have a slightly lower pressure there. That's a little bit more healthy for the heart and for the cardiovascular system. So systole, that is contraction of the muscles of the heart, while diastole is relaxation. And the valves open based on the differences in blood pressure on either side of the valve. So if we're looking, let's say, I'm just going to draw the left side of the heart. Here is the left atrium with a valve. Here is the left ventricle with a valve. The valve is either open, so here we see it open. I'm doing a terrible job of drawing. Sorry about that. It's really messy. And I'm going to do a dotted line with it closed. Okay, so there's the valve closed. So it all depends on the pressure on both sides of the valve. If the atria are in systole and they're contracting and they're pushing blood through, then the blood pressure inside the atria is going to be higher than the blood pressure inside the ventricles. And so that forces blood through the valves and it pushes the flaps apart. So high pressure here and low pressure here is going to open the valves. On the other hand, when we begin to generate high pressure here in the ventricles, then the balance shifts. So as we generate more and more pressure, then the relationship is that there's higher pressure in the ventricle, lower pressure up in the atria. That high pressure sneaks around behind, so we get blood pressure behind the valves, behind the flaps of the bicuspid valve here, in this case, or the mitral valve. And that high pressure closes the valves so that they go into this, into what we're seeing here. So now I'm going to draw them very dark. So now we see the valves closed because that high pressure has pushed them closed. So it's all on the differences in the blood pressure on either side of the valves. So if it's high pressure on the downstream side of the valve, that's going to close it. If it's high pressure on the upstream side of the valve, then that's going to open it. So, and if you recall, we've got those chordae tendineae here and a papillary muscle at the base of each of the chordae tendineae. And when they get pulled on, so as we tug on the papillary muscle with the chordae tendineae as they close, they respond reflexively with a contraction that helps to pull the chordae tendineae slightly tighter, so we get a little tension developed between the push being provided by the blood pressure and the pull being provided by the papillary muscle, and that prevents prolapse. If we didn't have this pull happening here, then the valve could flutter back into the atrium and we'd see prolapse. And specifically, the prolapse that's most likely to occur is mitral valve prolapse, because there's only two flaps there, and so each flap experiences relatively high pressure, especially when we compare it to the tricuspid valve over on the right side of the heart. Okay, so valve opening and closing is controlled by the differential pressure, the pressure differences on the upstream and downstream side of the valves. So when we're watching, so if we're looking now, I'm going to draw the whole heart with atria above, ventricles below. So a cardiac cycle then looks like, first we see the atria contract, so they constrict, that pushes blood down into the ventricles, and then the ventricles contract, which pushes the blood up and out either through the pulmonary trunk or through the aorta. So a single cardiac cycle involves contraction of the ventricles that pushes blood down, and then I said ventricles, I meant contraction of the atria. I'm going to make this drawing really horrible too. So contraction of the atria that pushes blood down into the ventricles, followed by a contraction of the ventricles that pushes blood up and out through the aorta and through the pulmonary trunk. So there are also valves at the pulmonary trunk and at the aorta. So if we draw those, here's the pulmonary trunk coming out, and then it divides into the right and left. Pulmonary arteries. Come on. Here's our pulmonary arteries. There's a valve here that it's shaped like this. There we go. And if we look at the aorta, it's got the same kind of a setup. Here's the aortic arch, and it has in it, right there at the mouth of the aorta, we see a valve. And so, let's see, they're connected to, here's the right ventricle below, and here's the left ventricle below. When the ventricles contract, pressure builds, and that opens up the valve, and so blood flows out through the valve, and the blood is so forcefully pushed out that the aorta and the pulmonary trunk, they're somewhat elastic, and so they kind of expand with that additional pressure. They get a little expansion here as they take on this big slug of blood that's going in. So they expand a little bit, as the ventricles begin to relax again and we get lower pressure here in the ventricles, then that expansion snaps back to its usual place, and that's what closes the pulmonary valves and the aortic valve, because the pressure pushes down on the cusps of the aortic valve and the pulmonary valve and causes them to bang shut again. So once more, high pressure on the downstream side will close the valves. High pressure on the upstream side inside the ventricle will open the valves. So we can hear these heart sounds as the valves are closing. And there are two primary heart sounds. One, it sounds 1, S1 and S2. S1, for you nurses, we're not going to use this terminology, but S1 is lub, S2 is dup. And the lub sound occurs as, I'm going to draw the heart again here, atria, ventricles. So here we have the AV valves. Lub is the snapping closed of the AV valves as the pressure builds inside the ventricles. Dup is the snapping closed of the pulmonary valve and the aortic valve as the ventricles, so as the ventricles build pressure, the AV valves close. As the ventricles release pressure and begin to go into diastole, then the aortic and pulmonary valves close, producing the dup sound. So lub is the AV valves closing, dup is the aortic and pulmonary semilunar valves closing. Okay? And auscultation, that just means listening to body sounds. So auscultation of the heart is done with a stethoscope. We can listen to vowel sounds. That's also called auscultation. Okay, so we see a little bit now about how the cardiac cycle goes, with first atrial systole, followed by ventricular systole. But how do we control this? What is it that tells the atria it's time for you to contract? And what is it then that tells the ventricles it's time for them to contract? So I'm going to do one more drawing of my heart. So here are the atria, here are the ventricles. This time we're going to pay more attention now to the muscle of the heart, because cardiac muscle cells are branching. So if you remember from back when we were looking at tissues, a single cardiac muscle cell is relatively short. It's striated. It has a single central nucleus, and they branch so that cardiac muscle cells connect to multiple other cardiac muscle cells. And they connect here at the intercalated discs, which are connections that include gap junctions. That's a kind of intercellular junction that allows ions to flow quickly and easily. And because ions are flowing, it allows that action potential signal to contract to spread very quickly from one cell to the next. And because the cells branch, there's all of these interconnections between our branched cells. So if this cell gets a signal to contract, it spreads it very rapidly to this cell and this cell. And so every other cell that this one is connected to is also going to get the signal. Every cell that this one connects to, and it connects both forward and backward, is going to get the signal. So you can see that this spread of the signal, it goes very rapidly, like fire, across any cells that are interconnected to each other. Those interconnected cells are referred to as a syncytium. Syn means, S-Y-N, that root, means together, cells, CYT. So these are the cells that are connected together, a syncytium. And so they all act in unison. They spread the signal to contract, and that signal is spread in a very rhythmic kind of way. It's spread in a very smooth way across all the cells that are interconnected. And all of the atria cells, so all the cells in the walls of the atria, are connected into what's called the atrial syncytium. And all the cells in the ventricles are connected together in what's called the ventricular syncytium. But they're insulated from each other by a firewall, you can think of it as of connective tissue, so that the cells of the atrial syncytium will not stimulate contraction in the cells of the ventricular syncytium, and vice versa. So all of the cells of the atrial syncytium, they're connected together, and when one cell contracts, all of the cells will contract. And the same with the ventricular syncytium, but there's almost no connection between the two, so that we don't accidentally trigger the ventricles to contract just because the atria are contracting. Okay, so how do we get the signal then from the atria? How do we generate the signal? How do we get the signal from the atria to the ventricles? That's the role of the cardiac conducting system. And the cardiac conducting system is a set of non-contracting cardiac muscles, cells. So they are cardiac muscle, but they don't contract. Instead, they just pass along the signal to contract. And there are two kinds of cells that are found in the cardiac conducting system. There are node cells. These are cells that can generate their own depolarization signal spontaneously. Remember, depolarization in a muscle cell is the signal to contract. It's the ions shuffling, the sodium ions flooding into the cell, followed by the potassium ions flooding out, and then the resorting of the sodium and potassium ions to the appropriate sides of the cell again to reset the cell. That's one of those depolarization events where there's the rising phase and the falling phase, and so we see depolarization, repolarization, hyperpolarization, and return to rest. Okay, so these node cells can generate on their own without any control or any being told to do it by the brain. So they do this all on their own. So they generate the depolarization. The conducting cells then distribute that contracting stimulus from the nodes and out to any cardiac muscle cells that are connected to those conducting cells. Okay, so two kinds of cells. The ones that generate it. We talk about the node cells in kind of common words as the pacemaker of the heart. So there's the SA node, the sinoatrial node, that acts as the primary pacemaker of the heart. And they depolarize spontaneously about 70 times every minute, between 70 and 80 times a minute. Where there's also a second set of node cells called the AV node. They generate their own depolarizations as well, but at a much slower pace. Okay, so the SA node, I kind of got ahead of myself there. That's a cluster of these node cells that are found in the right atrium near the opening of the superior vena cava. So I want you to notice that's a change in your notes. I had somehow gotten the wrong idea and put the wrong thing in your notes. So change that to fix your notes. So it's a cluster of node cells there near the opening of the superior vena cava. And it produces 70 to 80 rhythmic action potentials, boom, 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 slightly faster than 1 per second. And those rhythmic action potentials then spread across the atrial syncytium. And as a result, we see the atria go into systole. They contract. There's also an AV node that's down near the wall between the atria and the ventricles. And it produces much slower action potentials. It's part of the atrial syncytium. And so it generally receives the much more rapid SA node action potentials, and when it receives them, then it gets blocked from doing its own 40 to 60. So if it receives an action potential, then it will respond to that action potential and not to its own node, just node generating thing. So usually in a well-functioning heart, the AV node does not generate its own action potentials independently. It just picks up the SA node's action potential. And it holds on to that. It delays very briefly that impulse to contract, and then it is the break through the firewall, and it transmits the signals to contract down to the ventricular syncytium. Okay, so here we see the SA node again. It generates a signal to contract that spreads down and across the base of the heart, and it forces the blood. It starts the contraction up here at the topmost region of the atria, and it forces the blood downward toward this opening between the atrium and the ventricle, forcing open the valves of the AV valves there. It also triggers then the AV node, and the AV node holds onto that signal to contract, and then it sends it down the AV bundle and through the firewall, through the connective tissue that separates the atria from the ventricles. Now, we don't start our ventricular systole, we don't start it here at the superior portion of the ventricles. Instead, we send that signal through the AV bundle and then down the right and left bundle branches. Now, these are insulated, just like a neuron is insulated, and they prevent that signal to contract from leaking out, and instead the signal travels all the way down to the apex of the heart, and then right up the walls, kind of goes laterally along the walls of the heart. And it's only when the signal gets to these fibers that branch off of them, and again, they're non-contracting cardiac muscle cells that are called the Purkinje fibers. They don't have the insulation, and that begins the signal to contract. So note that we begin the signal to contract for the ventricles here at the apex of the heart, and that signal spreads upward so that we get a contraction that begins at the base of at the apex of the heart and pushes up, and then up here, up and out through the aorta, and up and out through the pulmonary trunk and into the right and left pulmonary arteries. So we can't, because of where the location of this slice is, we don't see those blood vessels, but this is the space where we would find the valves. Here's the space where we find the valves for the pulmonary trunk. This would be for the aorta. Okay, hopefully that makes sense now. So we initiate the atrial contractions from the top, from the base of the heart downward. We initiate ventricular contractions from the apex of the heart upward, and up through the pulmonary trunk. And again, I think I made this point earlier, but I'm going to make it again now. This is where the book makes it. The muscle fibers are arranged so that the grain of them spirals from the apex of the heart upward, and then here at the surface there's another layer of them that spirals again upward. And so as the ventricles contract, they don't just squeeze inward, they also kind of pull the apex of the heart upward as they contract too. So there's a little twist then as the heart contracts, and that really very efficiently wrings or squeezes the blood out of the chambers of the heart, the ventricles of the heart. Okay, so the last thing in this section of the lecture is about electrocardiograms, ECGs. They were originally called EKGs because they were a German invention. Cardio in German is spelled with K. But I think they're now more commonly called ECGs in our hospitals or places. So these are a way to graphically see what's happening with the electrical activity of the heart. So we can see the depolarization and repolarization of the changes in electrical activity that's going on. I am not going to test you over anything beyond this on electrocardiograms because I figure you're going to get really good instruction. If you wind up working with a cardiologist, you're going to get really good instruction about how to read EKGs or ECGs, and it's probably better than I would be able to do. So we see, though, the waves have names. They're the different peaks and valleys have names. So there's the P wave that's there at the initiating zone of at the beginning of a cardiac cycle. There's the QRS complex. That's the big spike upward involves that QRS complex. And then there's a T wave. I've given you a lot of notes about that in your notes package. But the bottom line is that we use these ECGs to detect cardiac arrhythmias. So that's a non-rhythmic contraction of the heart, and they help us to understand better how the electrical function of the heart is occurring. Okay, so I think that's all I got to say about ECGs, and that brings us to the end of this lecture. So if you have any questions, you know how to contact me, and please do it.